This is a recap of activating your anti-aging body. Part 3, the immune system, innate and adaptive, given at the Food Co-op, Taos, New Mexico. A recap of what we have learned before is that cells only live a short time and are constantly being replaced by new cells. An example would be your stomach and intestinal cells only last a few days. The lung alveoli, 8 days. The skin, maybe 10 to 15 days. Liver cells, only 6 months. Heart cells, 10 to 15% per year. White blood cells, only a few hours to maybe 30 days. Red blood cells, for most adults, between 90 and 120 days or less. So that's it. All this regeneration comes from stem cells. Stem cells are formed in the flat bone marrow of adults. Mechanotransduction is the mechanism for cellular signaling to grow adult stem cells, i.e. making new blood, white blood cells and red blood cells. And also that's the mechanism to produce bioelectricity. All cells that are alive have bioelectricity, and dysregulation of that bioelectricity produces cancers. The immune system is certainly highly regulated by the lymphatic system, and lymph fluid can only be moved by muscular contraction. Psychoneuroimmunology affects all of our cells, particularly the vagus nerve, singing, gargling, music, etc. 2,500 years ago, the Chinese promoted the idea that there's Jing, which I believe is the current understanding of a stem cell, Qi, which is our bioelectricity, and Shen, which is the consciousness, and the effects of psychoneuroimmunology on those on our body. So obviously, Tai Chi and Qigong are the best form of exercise. Weightlifting and aerobic exercise are things that actually produce free radicals that will harm the body. Only 10% of all illnesses is hereditary, and 90% of all of our doctor visits are truly only stress-related. I say only, but it certainly indicates that the cells are affected through the epigenetic changes because of our stress factors. Our immune system has two different types, the innate or non-specific immune system and the specific or the adaptive immune system. All this involves the quick unfolding of both B cells and T cells, and we'll explain what they are. T cells are precursor cells that come from an adult stem cell made inside the bone marrow. B cells are also made inside the bone marrow. The T cell moves from the bone marrow into the thymus, and from the thymus it then begins to circulate throughout the body. The B cells move directly from the bone marrow into the bloodstream and begin acting with an antibody response as quickly as possible. White blood cells are also made in the bone marrow. White blood cells are what fights infection and are the most important part of your immune system, but it only makes up less than 1% of your blood. There are three types of white blood cells: granulocytes, monocytes, and lymphocytes. The granulocytes, these granulocytes are neutrophils that help fight bacteria and fungal infections. The basophils are your body's immune response. They contain things that help the blood clot and release histamines, and also this immunoglobulin E, or IgE. There are 4 types of IgEs, or immunoglobulins: IgE1, IgE2, IgE3, and most importantly, IgE4, which is pretty much a autoimmune response that actually harms your body. But we can get a little bit more into that later. Eosinophils help fight infection caused by parasites. So all these are part of a white blood cell, and they're part of the granulocytes that are types of white blood cells. Next, there's the monocytes. The monocytes break down and remove foreign organisms and dying cells for your body. They are the macrophages for the blood. Macrophages engulf and digest pathogens such as cancer cells, microbes, cellular debris, and foreign substances, which do not have proteins that are specific to healthy body cells on their surface. Macro simply means large, phages is eaters. Macrophages is Greek for large eaters. They're basically floating around in your blood and they're cleaning up all the mess that's in there. Lymphocytes are made also, obviously, in the bone marrow, and their T cells are matured in the thymus. And there are two major types and a third regulatory type. The cytotoxic T cells are sometimes also known as CD8 plus cells because they have a CD8 receptor on their membrane. Helper cells are also sometimes called CD4 plus cells because they have a CD4 receptor on their membrane. Unlike the cytotoxic T cells, the helper cells don't kill cells directly. They call for the other C cells, the CD8 cells, to come and destroy whatever needs to be destroyed. The regulatory cells play a role to make sure your immune system doesn't attack yourself. These cells reduce the activity of the other T cells when necessary, and they most importantly prevent your body from attacking healthy cells. They regulate it. Now, B cells play a variety of roles in the antigen presentation ability through cytokine and chemokine productions. The innate immune activation of B cells may play a beneficial role through the generation of natural cross-reactive antibodies. By maintaining these high B cells, there's memory involved, and the exercise functions that may provide protection against autoimmunity problems. They're also, meaning these B cells are also part of the adaptive immune system, specializing in generating a protective high affinity antibody for a particular response that it doesn't recognize. It appears that even memory B cells belonging to the adaptive arm of the immune response can also be regulated through the innate immune receptors in an antigen-independent fashion. So these B cells are very important. They actually are there. They don't have to be trained to know what to do. They recognize things in your body, and they begin to attack them and remove them if they don't believe they're beneficial for you. They also can remember things that you had in the past and act quickly against them. There are different types of lymphatic tissues, and importantly, there's a system of lymphatic tissues called MALT, or mucosal-associated lymphatic tissues. NALT is a form of MALT. So NALT is cells that protect your nose and upper respiratory tract. BALT is a form of MALT, and it protects the bronchial region. And GALT is a form of MALT that protects the lining of the gut. Here's a little photo of what that looks like for the MALT, GALT, and NALT. You see these different aspects of the body and how the immune system is using these particular secondary lymphatic tissues and organs to help keep you strong and healthy. Here's a little video that explains a little bit more about the immune system and how it works. Let's take a look and see a little bit how they explain this.
In this animation, we want to show the human immune system with images never seen before. We will learn how macrophages as well as other cells and mechanisms protect our organism from death every day.
Macrophages are circulating in your blood. They're going to attack them and take out.
Our skin, like the bark of a tree, represents valuable protection. However, the skin is also home to countless microorganisms. Among other things, they are nourished and protected by an oily fluid called sebum. Sebum is produced by sebaceous gland.
Okay, this sebum, it's what kind of makes your skin glow. So when someone has a really healthy immune system, you'll see the sebum, or a nice glowing factor in their body. And that glow is part of that the substance that's on this basically a surface of the skin and just barely below it.
Sebum also contains antimicrobial elements that can kill microbes. If they are harmful or too many microbes on the skin, the sebum is composed in such a way that these microbes are killed. This creates a harmonious coexistence between skin cells and microbes, a kind of symbiosis. These microbes also keep one another in balance and kill pathogens that can be dangerous to our cells. The protective layer of hollow organs, such as the nose, mouth, and trachea, is a mucous membrane. Mucous membranes possess cilia. Cilia move back and forth in a wave-like manner. This allows them to move mucus, to which foreign substances adhere, upward into the oral cavity.
That's your
Mucociliary clearance is not sufficient. An attempt is made to achieve clearance of unwanted particles through coughing. This is done by inhalation, followed by a sudden exhalation, so that foreign substances are removed from the lungs and trachea.
The bolt, or
the bone marrow has stem cells. It is the birthplace for many different immune cells, which we also call leukocytes. Stem cells divide to form daughter cells, asymmetric or symmetric. Symmetric cell division results in either two stem cells or two progenitor cells, as seen here.
So where these white blood cells come from?
The progenitor cells divide again and again.
Stem cells inside the bone marrow.
Thus, a single stem cell becomes many immune cells or leukocytes. A common immune cell is the neutrophil. Some of the neutrophils remain in the bone marrow, while others make their way into the bloodstream. Our neutrophil belongs to the so-called innate immune system and moves through the blood vessels. During inflammatory processes, the endothelial cells of the blood vessels have special receptors. Thus, initially there is weak adhesion, then strong adhesion, and finally the granulocyte goes through the endothelium. This process is called diapedesis.
That goes through the outer layer of the cell.
The path through the connective tissue has already been made by other neutrophils. Our neutrophil only has to follow cytokines, that is, signal substances of other cells, a process called chemotaxis. There they are. Bacteria everywhere. They produce toxins and thus harm the cells of the organism. A neutrophil's job is to eat these bacteria. This eating process is called phagocytosis. In addition, some of the neutrophils produce certain cytokines that attract other granulocytes.
These phagocytes attach and they eat things that are poisonous, particularly the bacteria, to get it out of the body. It starts to break it down into particles that are either
other cells of the innate immune
the
system are macrophages.
Macrophages are the big eaters.
They arrive on the scene before neutrophils, phagocytose bacteria, and produce a signal substance, tumor necrosis factor. It suppresses the feeling of hunger, causes fever, stimulates leukocyte production. Another leukocyte type is the T cell. T cells possess receptors. What do they need them for? Macrophages are professional antigen-presenting cells. They have the ability to present antigens via certain proteins on their surface, and this initiates certain processes. Some T cells can use their receptors to recognize exactly those antigens of the macrophages for which they have been trained in the thymus.
They literally look for a match in how the cell
Let us take a
surface
closer look at this.
looks like.
T cells are also formed in the bone marrow. They then migrate via the bloodstream to the thymus. Because they mature in the thymus, they are called T for thymus cells.
Created
In the
bone
thymus,
marrow.
which is one of the primary lymphoid organs, T cells form antigen receptors. Afterwards, they usually migrate to secondary lymphoid organs, such as the lymph nodes and the spleen. In the lymphoid organs, naive T cells and dendritic cells meet. The dendritic cells have already had contact with a pathogen and present the antigen on the surface. They are also professional antigen-presenting cells. The T cell scans the dendritic cell for the antigen for which the T cell was trained in the thymus. If it finds the antigen, T cell activation begins and, among other things, clonal expansion occurs. The cell reproduces itself thousands of times. These cells respond to a specific pathogen or antigen. Then, some T cells start their journey and migrate to other tissues. Once there, the cytotoxic T cells scan other cells with their receptors to look for cells that have the specific antigen on their surface.
They find them, they're going to attach, and they're going to try to destroy them.
If they find their antigen in a cell, they know that a pathogen, for example, is in the cell. The cytotoxic T cells begin to induce apoptosis. The cell's debris are eventually eaten by macrophages and new cells form.
Those big eaters come over and take up the waste product, further break it down, dispose it through the body.
There are not only cytotoxic T cells, but others as well. T helper cells, memory T cells, regulatory T cells, and others. T helper cells work hand in hand with B cells. B cells possess receptors like T cells. Like T cells, B cells also originate in the bone marrow. They then search for their specific antigen in the lymphatic system or in the bloodstream. If the B cell finds its antigen with its receptors, either activation occurs immediately or the B cell moves into the lymphoid tissue where T helper cells wait. T helper cells, as we've seen before, were also trained for one specific antigen. For this reason, the B cell presents its antigen on the surface. This allows the T cell, with its receptors, to scan the B cell. If it is the antigen for which the T cell has been trained, it gives the starting signal for the division and maturation of the B cell.
The B cell begins to change, mature, to be able to attack a particular thing that's in your body.
As with T cells, clonal expansion occurs. Thus, thousands of new B cells or plasmablasts and plasma cells are generated from a single B cell. Memory B cells are also produced, which will spring into action in the event of another subsequent infection. During acute infection, plasmablasts and plasma cells immediately begin to produce antibodies, and they release these antibodies into the bloodstream. Antibodies are also often called immunoglobulins.
The IgG 1, 2, 3 go out into the blood looking for specific things to destroy and to remove from your body.
If the toxin, the bacterium, the antigen is in the blood, the antibody just produced by the plasma cell can bind to it. And this way, for example, a toxin can be rendered harmless and an antigen can be marked so that macrophages can recognize it more easily and phagocytize it more quickly. This process is called opsonization. All immune cells have a stem cell as their origin. It develops into progenitor cells and eventually into the immune cells just shown. Other important immune cells are natural killer cells and platelets.
Okay, so you get a general idea here of what's taken place. So we have specific things that happen in the body always in response to anything harmful coming into the body. These key things, these B cells and these T cells, these lymphocytes, they're made in your bone marrow. So what we talked about in our other lectures is that the flat bones in your body is where the adult makes these new blood and where this immune system originates. So the flat bones are your wing bone, the sternum, the sides of the ribs, and the hip itself, the flat part of the hip, the iliac crest. So here's where we're making all of this new blood, all these new white blood cells that have all these T and B cells in it, and they're floating around in our blood, and they're trying to clean it out and take toxins out of our body. But there's also the lymphatic system that we're going to get into a little bit more. We talked about the lymphatic system in our first class. And the lymph system is where we have lymph nodes where B and T cells are holding in their body. And those lymph nodes are places like behind the knee, in the groin, underneath the armpit, in the neck, the bottom of the stomach, between the belly button and the sternum is where a lot of them collect from your legs, called the cisterna coli. Now we're going to talk about this in a few more minutes. So that's all happening through the bloodstream and what's happening to a cell. But now we're going to talk a little bit what happens about the immunology inside the gut system. So both in your stomach and intestinal tract. So let's take a look at this one and see what's happening with that. That same villi we saw earlier, where that happens to be inside the body, not just in the lung area. It's along the digestive tract.
The human gut can be the scene for devastating conditions. Patch is a mix of immune cells including T and B lymphocytes, macrophages and dendritic cells. A key function of the Peyer's patch is the sampling of antigens, in this case mostly bacteria and bits of food. To facilitate this, the Peyer's patch has a much thinner mucus layer as well as specialised phagocytic cells called M cells, which can transport material across the epithelial barrier via a process called transcytosis. Finally, dendritic cells are able to extend dendrites between epithelial cells to sample antigens that are then broken down and used for presenting to lymphocytes. Sampling antigens in this way typically results in so-called tolerogenic activation, where the immune system initiates an anti-inflammatory response. With their cargo of antigen, these dendritic cells then traffic to the T cell zones of the Peyer's patch. Here, the Tregs secrete a molecule called IL-10, which exerts a suppressive action on immune cells within the lamina propria and upon the epithelial layer itself. IL-10 is therefore critical in maintaining immune quiescence and preventing unnecessary inflammation. However, a breakdown in this process of immune homeostasis results in gut pathology, and when this occurs over a prolonged period and in an uncontrolled manner, it can lead to inflammatory bowel disease. Chemical, mechanical, or pathogen-triggered barrier disruption, coupled with particular genetic susceptibilities, may all combine to set off inflammation. Epithelium coming into contact with bacteria is activated, leading to bacterial influx. Alarm molecules released by the epithelium activate immune cells, and Tregs in the vicinity scale down their IL-10 secretion to enable an immune response to proceed. Dendritic cells are also activated by this environment and start to release key inflammatory molecules such as IL-6, IL-12, and IL-23.
Okay, so you can see the similar response takes place either on the surface of the skin, inside the blood, and now we're seeing it inside your digestive tract as well. A key component of all of this is the lymphatic system itself. So in front of you now, you have this showing of where the lymphatic vessels are. And you see how they start way on the bottom here from your feet, and they come up. You have behind your knees, these are called the popliteal glands here. Then you have these in your inguinal area, the inguinal lymph nodes that are here. And all of these on the legs and the abdomen all drain into the cisternal coli right here. The cisternal coli then has to move this up to the left jugular vein. So as it moves up into the body, it's going to drain it into the left side of your body, into the left jugular vein. And that's going to then clear this into the venous system, where it's going to be recycling the parts out that are harmful and then recycling the parts that can be used back into the body again. The key thing is that your legs and your hips and your waist, that all drains into the left jugular vein. Only the right side, with the right arm and the right part of the chest, drains into the right part of the subclavian vein. So the key thing is that the lymphatic vessels do not have a pump for it. So the lymphatic vessels are only moved by, there's a little bit of smooth muscle cell around the vessel itself, but most of it is moved by the skeletal muscles that have to squeeze and release in order to get the lymphatic fluid moving from the lower part of your body all the way up and eventually drain into the vein in the neck. If you're sitting around not doing anything, you're not able to drain the lymphatic vessels properly, and toxins literally are building up in your body. And this whole thing that we saw that takes care of your body and protects your body from your immune system is not going to work unless you physically move. So you not stretch, you have to physically squeeze and release the skeletal muscles. So things like walking would work, certainly all Tai Chi and Qigong would work. And also you have to make sure that you drain it by moving something, by bending forward or back at the waist to get the fluid to leave from the cisternal coli up into the left jugular vein. This is what looks inside of a lymph vessel. So you see there's flow coming in from the capillaries into the lymph vessel. And then there's these little check flow valves. So the check flow valves are designed to show you that when the fluid inside the vessel squeezes, it can't flow back. So these are like check flow valves or backflow valves, where it will stop the fluid from flowing back. It'll continue moving in one direction, which is up, until they get to be drained again in the vein. So like the veins, the valves and the skeletal muscles pumps are important to ensure the return of that fluid properly. All right, we're going to go into a little bit about vaccination. So inoculation was something that actually was initiated at least as early as 1000 AD in China. So this is a picture of a person who was blowing a smallpox pus or smallpox scabs that have been dried into the nasal cavity of a young person here. And because it's sent in there, the body's going to recognize that smallpox, and it's going to create a response to that poison that just went into your body. So those B and T cells are going to look and say, oh, if I ever get the live part of this, I'll know what it looks like and destroy it right away before it can build up any colonies. So this practice was not only in China, but also in India and in Africa. And in India, they sometimes put a little cut on a person's arm or their hand, and then they put these dried scabs or pus into the cut, so it began to circulate directly into the bloodstream. Later on in 1796, milkmaids who had developed cowpox, which is like smallpox but for cows, they developed some lesions on their body, and they found out later that those who developed those cowpox lesions were resistant to smallpox. So he took material from the cowpox on the hand of this milkmaid, Sarah Nelms, and inoculated a young boy with it. And his resistance to smallpox was proven, and so he began to extend it to other children. So in 1796, we developed the word vaccination from the word vaca, which means cow. So we were inoculating ourselves from smallpox by 1796. But by certainly 1000 AD in China, India, and Africa, they were already doing it. Now, the CDC says that vaccines work by imitating an infection. So from that picture we just saw and from that information we just learned, the vaccines will look at the presence of a disease-causing organism in the body, and it'll engage a natural defense against it. The active ingredient in all vaccines is the antigen, the name for any substances that causes the immune system to begin to produce these antibodies. In a vaccine, the antigen could be the weakened or killed bacteria or virus, like we saw in those that slide, bits of the exterior surface or the genetic material of that pathogen, or the bacterial toxin treated to make it non-toxic. And this is the way vaccines all used to be created. But along came COVID, and we changed the way we make vaccines into something new. I'm going to talk about that in a minute. To determine how effective a treatment is, particularly for vaccines or for any treatment modality, there's certain statistical terminology that's used to give an example of the benefit you might receive from the treatment method. So in 2010, scientists got together and said, we need to agree to something called the consort statement. It stands for the Consolidated Standards for Reporting Trials, clinical trials. It was a 25-item checklist. But for you and I, the most important thing on that checklist was to look at what is the absolute risk reduction, what is the number needed to treat or number needed to vaccinate in order to stop one infection, what is the risk ratio, and what side effects might there be. So informed consent was designed to be you as the consumer of medical products need to know what these things are. The key part is the absolute risk reduction, or also called a risk difference. It's the most useful way of presenting research results to help you in your decision making. So we're going to show an example where the absolute risk reduction was 8% because 20% of people took a measure and they were prevented from getting whatever the thing was. 12% didn't, so they had that difference between the 20% who were basically the placebo group and 12% who were the control group. And the 8% showed that that was the absolute risk reduction for that model. So this means that if 100 children were treated, 8 would prevent it from developing a bad outcome. Another way of expressing it is looking at the number needed to treat or vaccinate. So if 8 children out of 100 benefit from a treatment, the number needed to treat for one child to benefit would be about 13. And that's found by taking the absolute risk reduction divided by 100, and that gives you a percentage. And that is what your number needed to treat is. All right, here's another example. Comes from the National Library of Medicine in a book called Smart Health Choices, Making Sense of Health Advice. And it explains in chapter 18 specifically the relative risk, the absolute risk reduction, and the number needed to treat. So here we're looking at it again. So we talk about this absolute risk reduction. Suppose there's a well-designed randomized controlled trial in children with a particular disease and found that 20% of the control group developed a bad outcome compared with only 12% who received a treatment or a vaccine. Should you agree to give this treatment to your child? Well, without knowing more about the adverse effects of the therapy, it appears that this might be a good thing. But is it meaningful statistically based on the whole potential population of different variables? So, well, how do you find that absolute risk reduction? Or the risk difference is to do this subtraction between the number that got it in the control group and those that didn't. And that's where you had the 20% minus the 12% to get you 8%. Again, so that meant that if 100 children were treated, only 8 would be prevented from having a bad outcome. Okay. In a research paper on perspective of clinical research says the common pitfalls in statistical analysis is looking at absolute risk reduction, relative risk reduction, and the number needed to treat. Again, the absolute risk reduction or risk difference represents the proportion of patients who are spared the adverse outcome as a result of having received the experimental drug, vaccine, whatever, rather than the control therapy. Okay, this is from training in looking at things from a point of view based on whether or not a drug would be beneficial. Again, we have that absolute risk reduction, which is the custom rates between the control group and the treatment group. The number needed to treat is another way to express that absolute risk reduction. The number needed to treat is the most intuitive statistics to help answer the question of whether or not it's a beneficial thing. So if the number needed to treat is between 2 and 4, then there's an excellent benefit that the antibiotics for infection would work. If the number needed to treat was between 5 and 7, it's associated with a meaningful health benefit, as in some antidepressants. While a number needed to treat at 10 or greater than 10 is most associated with a very small net benefit. And it talks here about using statins to prevent heart attacks, because statins carry a lot of other high-level side effects with only a limited amount of benefit. So the number needed to treat is much higher there. So let's look at some basic things that the Cleveland Clinic has told us about getting a shingles shot. So when you have shingles, it shows you here's how many people were in the control group, how many people took place. And right here, the Cleveland Clinic clearly shows us the absolute risk reduction is 1.7%. The number needed to treat was 59. And it goes on, it says, if you had to look at the people who actually had then prevention of pain, and then this neurologia of pain, it only occurred in 27 of the people. And so that means that the absolute risk reduction to not have pain associated with taking the shingles virus was only 0.27%. And the number needed to treat to prevent one outbreak was 362 people. Now, you might wonder whether or not that's a beneficial thing to do when 1.7 from getting it percent only, and less than 0.27% to have it where you wouldn't have any pain if you got it. So that's pretty low numbers, yet they seem to be pushing vaccinations of these all the time. Now, you as a consumer have to decide what's beneficial for you, okay? And you need to know this absolute risk reduction number, the number needed to treat, the number needed to vaccinate. Okay, now, the theory of cognitive dissonance is this idea that there may be inconsistencies within our own mind where we're giving thoughts, words, behaviors that seem to clash with my idea of something. When I learn new information that challenges a deep held belief I hold, or act in a way that seems to undercut a favorable self image, I'm motivated to somehow resolve that feeling of weirdness inside me and restore a level of cognitive consonance. So I not always resolve this cognitive dissonance. So I might look at things in a different way. So, example, if I learned that my eating habits really suck and it increases my risk of illness, I feel the tension between my preferred belief of myself and the idea that I could be in danger. So I might think, hey, this is exaggerated health warning. I really don't have to believe in that or do any changes. So I kind of fluff off what I know is something that's going against how I view myself. I see myself as being healthy. Why do I need to worry about these other things? Now, conversely, I could take the information that's kind of coming into me and I could decide to take a change in my behavior. I could say, hey, maybe I better start exercising. This cognitive dissonance also kicks in. Say you have a favorite politician who has done something that doesn't seem to be right. You could conclude that the charges against this person were invented by their enemies. Or maybe you might want to think about rethinking your support for that person because you've got new information. So you have to deal with how this idea you have of something and conflicting information comes into it and what you're going to do with that, because it does create confusion in our minds. Okay, so now we're going to get into this idea about genetically reprogramming you. What are the pros and cons of it? So remember I said about how vaccines used to be created? Well, now vaccines are being developed in a different way that I want you to know about.
Thanks to Brilliant for supporting this episode of SciShow. Check out brilliant.org/scishow to learn more. Two of the vaccines we have for COVID-19 have the distinction of being the first mRNA vaccines to see widespread use in humans. It's a vaccine technology that uses a type of molecule called RNA to teach our cells how to resist disease. So how do they work? And how are they different from the litany of immunizations you probably got as a kid? Let's take a look. All vaccines do one basic thing. They imitate a disease-causing agent or pathogen with the goal of ratting it out to your immune system. Then your immune system knows what to do if the pathogen ever shows up. mRNA vaccines do that too, but they represent a departure from the way we've introduced pathogens in the past. Most of the time, we've used a bit of the real thing, like killed pathogens or living ones that have been weakened, or just pieces of them, or
okay, just pieces of them or the actual pathogen itself. That's how our body then was able to respond to it and say, hey, this doesn't belong here. Let's get it out. Now an mRNA vaccine is different. They're going to be sending a signal inside your body, literally to genetically program you to make the spike protein. Let's watch.
even substances that bacteria just make. In any case, these bits our immune system can spot are called antigens. Each antigen has a shape unique to a particular pathogen. When you get a vaccine, your immune system sees the antigen and sounds the alarm. That triggers the formation of memory cells that remember the bad guys, as well as antibodies that disable the pathogen if it tries to invade in the future. Now, mRNA vaccines still accomplish that goal, but instead of pieces of pathogens or any other strategy we've used in the past, they contain messenger RNA, or mRNA for short. It works sort of like giving our cells a build-your-own antigen kit instead of the actual antigen. The end product is the same, but we've asked our cells to do the heavy lifting.
Okay, the end product is that our body's going to have to respond to a spike protein that we just genetically created within our body. So I want you to clearly understand that. They're literally are taking something into our body that is genetically reprogramming our body to produce a spike protein. So we're making the spike protein. It's not that we got it from nature or from a virus somewhere in the past, or from like the smallpox itself, or the measles, or the mumps. We're literally programming your body to make a spike protein by telling it to do it. And then the goal then is to have your body respond to the spike protein that you just told it to make. So your immune system doesn't get enhanced by the spike protein. You either have a strong immune system or you don't. All it does is make the spike protein.
That's because cells are kind of better at this stuff than we are. We can mess around with pathogens all day, but cells already know how to make antigens if we just supply the instructions. It's not the only way to design a vaccine fast, but it's a great tool to have handy when a pandemic comes along.
Okay, so here she's promoting this idea. It's a great tool to have handy when a pandemic comes along. So I want you to understand that this mRNA technology, she's going to say it's been around for a long time. It's been here trying to finally get approval, but it was approved on emergency authorization. And it didn't go through its whole list of looking at specific problems that may come up when you are reprogramming you. So when you're making this genetic change inside you to produce a spike protein, and that spike protein, you're hoping your immune system's going to be strong enough to fight it off and to destroy it. Let's take a look and see what they say.
So, how do you program a cell to make an antigen? Well, the simple version is you just borrow the machinery it's already using. See, most every cell contains a complete copy of our genome, the sum total of instructions it takes to make us, written in DNA. That information gets translated into proteins, which do all the actual work of being alive. Every protein has a job, specified in the DNA instruction manual. When it's time to make a protein, the cell needs to reference those instructions, but DNA remains in one spot, the nucleus of the cell.
So the DNA stays in one spot, the nucleus of the cell. But we're going to see here how the RNA is going to be able to come into the cell, and that's where the information is going to be genetically programmed into you to have your body create the spike protein.
Proteins, meanwhile, are made outside the nucleus. The solution is to copy out the instructions and send a message outside the nucleus. You might see where this is going. That message is made of messenger RNA, which contains a copy of the DNA instructions for use in protein making. The message exits the nucleus and makes its way to a ribosome in the cell. This piece of machinery reads the mRNA and translates the message into a protein. This is where the clever part comes in because we know the genetic code. So we can write our own message for the cell to translate. That's what.
So you can write a message for you to create a spike protein. So this is you messing with your genes to create a spike protein.
An mRNA vaccine is a coded genetic message from us to our cells for them to work out with their molecular decode.
And it wasn't us to ourselves. This is from a laboratory that says, oh, our human genome looks like this, and we can find this spike protein by looking at the spike protein's structure and makeup, and how we can trick our body into making the spike protein by having this adjusted mRNA come into the cell and then take the instructions that we literally programmed your body now to make for a period of time.
Decoder rings. Instead of an antigen, they contain the mRNA template to build an antigen. In the case of our COVID-19 vaccines, the message codes for an antigen called the spike protein. These spikes stick out of the virus, which uses them like a key to unlock and infect people's cells. When the shots go into people's upper arms, the mRNA molecules inside make their way into cells and to ribosomes, which are more than happy to do their job and translate the message. Our cells display the finished spikes on their surface. Our immune system spots them and starts taking action. From there, it's thought that things work pretty much the same as.
Okay, so from there, it's thought that they work pretty much the same, so that those T lymphocytes and B lymphocytes
Thanks to brilliant
have to come in and they have to find a way to destroy this thing they're making. So this is another way of looking at it. So here are these nanoparticles that are coming into the cell. It carries within information to create these little things here, which eventually become the spike proteins. The spike proteins will then exit the cell itself. And as they exit the cell itself, this is where the spike protein is demonstrating its particular architecture. So it looks a specific way. So your B cells come along and they see that and they attach to it and they begin to try to destroy it. Now, there's different exposures that we have on the different technologies with the mRNA shots. So how long will these instructions stay in your body to build the protein? Some say they were a few months. Current information I'm reading says it may stay for as long as 6 months to be continuing to producing that spike protein. But that's on the outside of the cell. Since we decided we're going to reprogram us from some factory somewhere, and who knows where they made them, you're relying on them to give the proper information to start restructuring your body to develop this spike protein. But something happens. We wanted to know what happened. So you, as a consumer, have a right to know. And so the COVID vaccine clinical trial data that showed what happened, like how effective was it? How effective is it to put this modified RNA inside of our body and have it change us to make a spike protein? And how effective was it in stopping the transmission of COVID? Well, when the scientists asked the FDA to release that information, when they asked Pfizer to release it, they said no, which is like kind of hard to believe. They just simply said, no, we're not going to give it to you. So this group called Public Health and Medical Professionals for Transparency sued the Food and Drug Administration. And here's where the particular order is. You can look it up. This is a PDF file you'll be able to look at. So when they filed this complaint saying, we need to see whether or not this was working or not, what is the absolute risk reduction? What's the number needed to treat or vaccinate? What are the risk problems that became associated with it? The FDA said, no, not only you're not going to see it, but we want to wait 75 years to publicly disclose it to anyone. Now, this is just mind-boggling that you, as a consumer, weren't able to see what this information was. Your doctor wasn't able to see what this information was. So let's look. First, the judge said, wait a minute. Open government is fundamentally an American issue. It's not Republican or Democrat. A popular government without popular information or the means of acquiring it is but a prologue to a farce or a tragedy or perhaps both. Knowledge will forever govern ignorance, and a people who mean to be their own governors must arm themselves with the power which knowledge gives. John F. Kennedy likewise recognized that a nation that is afraid to let its people judge the truth and falsehood in an open market is a nation that is afraid of its people. And particularly appropriate in this case, John McCain correctly noted that excessive administrative secrecy feeds conspiracy theories and reduces the public's confidence in the government. So the judge said, this is all the judge's direct words. He said the purpose of a Freedom of Information Act is to ensure an informed citizenry, which is vital in the functioning of a democratic society. And he gives the court cases that said that that's most important. Congress has long recognized that the information is only useful if it's produced in a timely manner, and that therefore excessive delays by the agency in the response is often tantamount to denial. So when needed, a court may use its equitable powers to require an agency to produce documents according to court-imposed timeline. So this judge on the 6th day of January 2022 said, produce the records and produce them now, not 75 years from now. Okay, so what did those records show when they started producing them? All right, the absolute risk reduction and the number needed to vaccinate. So if you look up here, this top line, it was the very first one that came out. The number needed to treat was 119 people to prevent one infection. The absolute risk reduction was 0.84%. And then later on, other vaccines were approved. And this shows you the specific absolute risk reduction. Most of them less than 1% or just about at 1%. And number needed to treat somewhere between 119, lowest was 86 or 82 down here, highest was 131. So that means that it wasn't effective in stopping transmission from the very beginning. Remember, it's the earlier thing we showed about if the absolute risk reduction was greater than 10, it was probably not very beneficial. This is 119 that the first vaccine was approved with. Now, what happened with the people who took it? Well, there was a group of people who took it. There was a double-blind, randomized controlled study. And this was one of the reports that was the FDA wanted to hide. It's the cumulative analysis of post-authorized adverse events. An adverse event is a potential side effect. So in this document, you'll see there are 1,540 different side effects in the vaccine group. And look at them here. Not only were there general disorders, but there are nervous system disorders, musculoskeletal system disorders, gastrointestinal disorders, skin and tissue disorders, respiratory disorders, but also infections, cardiovascular disorders, and 1,223 deaths were in the control group. That's how many people died in the control group after receiving this virus vaccine. Well, I don't know if we can call it a vaccine. After receiving this shot that created a spike protein inside of our body, so our body could fight that spike protein if the virus ever entered our body, or fight the spike protein because you got the vax in your body because it's making that spike protein, even if you don't have COVID in the first place. So let's look at that again. This again specifically tells you 1,223 deaths. This tells you what the age category it is. If you look in here at this line, 31 to 50 was the largest age group of these adverse events, followed by the 51 to 64 age group. And then it jumped to over age 75. So these are the systems that we talked about that after they received this vaccine or this spike-producing messenger RNA, this is what happened in the control group. This tells you what the absolute numbers were. All right, key things to remember here. On December 11th, 2020, Pfizer COVID vaccine was made available under emergency use authorization, meaning it didn't go through all the proper following of what happens after people are given it. The clinical trial data was not released until the 6th day of January 2022. But on September 2021, Biden Executive Order 14043 mandated the vaccine for all workers, all healthcare workers, all federal employees. Not one doctor was able to look at the clinical trials. Not one doctor was able to know what the number needed to vaccinate was. Not one doctor was given the information about the side effects. Not a single doctor had the knowledge to make an informed consent about whether the vaccine was actually truthfully effective and safe. Now, this is from the Associated Press. So they asked on January 25th of 2021, Remember, the data wasn't released yet. They said the Chinese Center for Disease Control, Gaofu, said he can't rule out negative side effects from the mRNA vaccine. It's the first time it was given to healthy people, and there are safety concerns. Now, the Associated Press said, oh, it was given to 70,000 people, and there wasn't anybody having any trouble with it. Well, this is on January 25th, but the clinical trial data wasn't released until January 2022, a year later. So they couldn't have known those side effects. All of these side effects you're seeing here, they were part of that vaccine group. Those were the potential difficulties that people were facing. And the news media is telling you, don't worry about it. It's not a big deal. Over 70,000 people were tested and it's safe. Trust us. Well, they didn't know it was safe or not because the data wasn't released. All right, now let's talk about a little bit further things here. This is from the peer-reviewed journal called Genes. It says mRNA vaccines, why is the biology of retroposition being ignored? Retroposition is reverse transcription, meaning it can change your DNA. So right now it was designed to be a part of RNA, but a messenger RNA or a modified RNA that went into your cell and produced the spike protein. This argument was being made that maybe it's possible for this mRNA type of vaccine to reverse transcribe into your DNA. So the person said, we urgently need experimental studies that would rigorously test for the potential retropositioning of the vaccines. At present, it seems like this issue is unresolved. Okay, now we're going to look at Professor Rudolf Janisch. He's a stem cell trailblazer from the Massachusetts Institute of Technology. He pioneered the field of epigenetics, which is meaning above the gene. We talked about that in our previous talk. It looks at how gene expression is regulated in the cell. Along the way, he created the world's first transgenic animals and laid the foundation for stem cell technology, which have become central tools for biomedical research. So he's going to talk about how, yes, the COVID virus does enter into your DNA. See the title up here, a permanent presence in our own DNA. So meaning the virus actually has gone into your DNA now. It did reverse transcribe. And he's going to talk about the dangers of what would happen if the vaccine, the message RNA spike protein, also if it went into your DNA in reverse transcription. So let's listen.
Line 1 is really expressed at a very low level. And people argued, well, how could this ever play a role in the disease? Well, it turns out that line 1 is expressed, is activated in aging, in cancer, and when you stress cells, for example, by virus infection, and cytokine exposure. And all these are risk factors for COVID-19. So we believe that the virus can integrate virus sequences, but we don't know what the relevance for the disease is. I will come later back to this. But I want to come to another issue, which is of major public interest. Namely, could vaccine RNA also integrate? And this is, of course, of concern to many, because if sequences integrate in your genome, they could cause mutations like cancer or could cause autoimmunity. So we don't know this. And so people are very concerned about this. And there's a lot of misinformation, as you're probably all aware, which really prevents people to take a vaccine. For example, people argue you might turn into a monkey if you get vaccinated. So the question, so what I showed you is that Virus infection produces internally lots of viral RNA, and that can integrate. There's no doubt. The question is, if you take RNA, same RNA, but not coming from an infection, but from a transfection or from vaccine, can this integrate?
Okay, so the question he's asking is, can the transfection or the vaccine integrate? We already talked about, and you can watch this on your own, that yes, the RNA does reverse, meaning it retrointegrates into the DNA. And let's take a look what happens. He says, if it does happen when the RNA or the messenger RNA comes into your body from the vaccine.
This really addresses this major question. So we looked at this. And again, the positive control is you're using the overexpression of line 1. That's just, and I showed you 3,000 copies per 1,000 cells after virus infection. After transfection with the same RNA produced in the virus, it's only 3. So it's 1,000-fold less. But this is the positive control. So look at the normal cells, and I showed you before, in normal cells, integration for virus infection is 1 copy per 1,000 cells. And of course, we didn't see anything in the RNA transfection. This means none, 1 or 0 means only it's 1,000-fold less, presumably from our positive result, than virus infection. So we believe it may be even less than that. So we believe that viral RNA does not integrate to any appreciated amount, but we have to, of course, really test directly vaccine RNA, not naked RNA, as we did, and we have to study the spike RNA, which is in a vaccine.
Okay, so he said we have to really study the spike protein and the spike RNA. But here on the bottom, you can see it says the vaccine and the spike RNA yet to be studied. So he says if it would go in,
and that is being done,
it would create these cancers. It would create autoimmune disorders. It would create and accelerate aging in your body. So that's the key thing to realize, that he made this study saying this talk, and you can find it on YouTube. Here's where the information's at. That he said when he was looking at it here, but not looking at the vaccine or the spike protein, it wasn't integrating. But let's take a look at what the new science showed. Okay, this is published in the current issues of Molecular Biology. Intercellular reverse transcription of Pfizer's BioNTech COVID-19 mRNA vaccine in vitro in human liver cell line. And remember that cell line 1 he mentioned? That's exactly where it went into. Their conclusion: our study is the first in vitro study on the effects of COVID-19 mRNA vaccine on human liver cell lines. We present evidence on fast entry into the cells and subsequent intercellular reverse transcription of BNT162b2 mRNA vaccine into the DNA. Reverse transcription is the process in which an enzyme makes a copy of DNA from RNA. So literally, those of you who took the vaccine have the potential of it literally changing your DNA. Now, the government's still telling you that that can't be done. I'm showing you this peer-reviewed research shows exactly that it has. Now let's see what this means to us. If you or someone you know is experiencing brain fog after COVID-19, scientists now have a possible explanation, and it might not bring much comfort. About 22% of people living with long COVID symptoms specifically reported this issue. And on average, 43 published studies. People who even had mild COVID illnesses can develop brain fog later. Why are researchers blaming the spike protein and not the whole COVID virus? As a part of the study, they found that the SARS-CoV-2 virus RNA in some people after death and not in others, suggesting that the virus does not need to be there to trigger brain fog. However, the spike protein does. So they also injected the spike protein directly into the brains of mice and showed that it can cause cells to die in the brain. They found no SARS-CoV-2 virus in the brain, the functional tissue containing the nerve cells and the non-nerve cells, but they did detect the spike protein there. So we're seeing that the spike protein that you allowed your body to make by having it be genetically programmed does cross the blood-brain barrier and does actually reverse transcribe into your DNA, all for less than 1% absolute risk reduction, coming with a host of side effects, including this. Well, people will say, well, I didn't die from it, and it kept me out of the hospital. Well, here's research that said that actually didn't happen. So the United States government stopped recording by vaccination status, but in Great Britain, they continued it. So here's what they're showing you, the number needed to vaccinate for the different age groups from 20 to 29 years old. In order to prevent even one infection, you had to vaccinate 169,200 people. To stop a severe hospitalization, you would have to have vaccinated 706,500 people who are between the ages of 20 and 29 years. And it goes down into the different years. You see the ranges here. The number needed to vaccinate to prevent COVID from developing was 1 in 700 people. So I gave this example when we talked about this less than 1% thing, or this 0.84%. Here in Taos, New Mexico, in Taos, we have about 6,000 people in the town of Taos. So if everybody in the town of Taos got vaccinated, that means that only 50 people would have been prevented from getting COVID. Only 50 people. That means 5,950 people would have still got it. If you look at the county, there's 30,000 people in the county. Of the 30,000 people, only 250 people might have been prevented from getting COVID. That means 29,750 of us would still have gotten it based on the clinical trial information. And it wasn't stopping hospitalization. And you're going to see next, it's not stopping death. In fact, the CDC, after being harassed by many people, finally agreed that there is a causal association between mRNA vaccines, particularly for Moderna and Pfizer's and biotech, and myocarditis and pericarditis. Myocarditis is inflammation of the heart muscles, and pericarditis is inflammation of the lining outside the heart muscles. Both of these occur at the same time. These conditions, inflammation occurs in response to infection of some other triggers. CDC has published case definitions for the myocarditis and pericarditis. Well, they say it happened in young people, but it happened in everybody. It has happened more in young people. So here's showing you there's a causal association, meaning that those things that we talked about, those adverse events that were recorded, well, the more we looked at them and the more the clinical trials were being observed, that information was indicating that it's causing clotting and inflammation inside the body, particularly heart disease. This is something that's found in autopsies recently, within the last 2 years, by embalmers. So as they're doing their embalming and as they're doing different autopsies, they're finding this strange substance that's developing after death. They don't know what it is. For some reason, the stuff inside is appearing there, and they have no idea what it is. Inflammation of some kind, but no one knows. Well, did it prevent deaths? There was 99.73% similar recovery rate between the vaxxed and the unvaxxed. At the end of 2021, the CDC stopped recording deaths by vaccination status. That speaks for itself. Okay, now we know that the information wasn't made available, that it was censored by the Biden's FDA. We then know that it took a court order to release the information. So when people tried to share online, they were prevented from doing so by Twitter, Yahoo, Facebook, Google, YouTube. They literally had the Biden administration officials calling people and telling them to remove the posts. And this is what I personally got. I got account warnings because my post went against the community standards. Take a look here. So I simply said, oh, those scientists filed the following lawsuit, and I gave what that was, nothing else. That was said to violate the community standards. Over here, I said, anyone can verify what I'm saying by going to the CDC morbidity and mortality rate and look it up. That was removed as a post. It violated their community standards when I showed you factual information. And not just me, everyone was showing people factual information. Well, not everyone, many people besides myself, and they were constantly being censored by these social outlets at prompting from the Biden administration. For some reason, Biden's FDA and the Biden government does not want you to know the truth about these vaccinations. Let's take a look at here in New Mexico. The UNM vaccination policy was everyone had to be vaccinated if you taught there or went to school there. But Dr. Christine Beato, the chair of the UNM's Health Protocols Committee, made this statement finally. Unfortunately, when the vaccine started coming out, we were told 92% of you, if you get vaccinated, you will not get the virus and you will not transmit it. We now know that that is not accurate. And even with vaccination, you will get the virus and you will transmit it. It was never accurate. It was never valid. You were misled. And on effective March 13th, 2023, University of Mexico no longer required the COVID-19 primary vaccine series or boosters for employees or students. Okay. That mandate is no longer based on the most up-to-date science. It contains some outdated provisions that have limited the outcome and hinder our ability to manage it effectively. It never was valid, ever. Okay, so why aren't we being told this? Why aren't information on the news? How are you and me going to learn about it unless we tell each other and start sharing things with each other? BlackRock and Vanguard are two of the biggest companies. Mostly they're weapons producers. And they own, you see, 18% of Fox News, 16% of CBS and 60 Minutes, 13% of Comcast, and Comcast owns NBC, NFC, CNBC, Sky Media. They own 12% of CNN. You see that they're owning the highest shares of the news media that we're relying on to give us information. And when they say we don't want you to know about something, believe me, the news doesn't tell us. They hide it from us, and they hid this information from us.
Vaccine is not just protecting themselves, but reducing their transmission to other people and allowing society to get back to normal.
We can kind of almost see the end. We're vaccinating so very fast. Our data from the CDC today suggests, that vaccinated people do not carry the virus, don't get sick.
Getting vaccinated and getting a booster shot when eligible can save your life and protect you and your family and friends from getting seriously ill and spreading infection. What do you think the probability is? 80%? Personally, I think it's 100%. I think that there's a reduction in transmission.
Essentially, vaccines block you from getting and giving the virus.
If enough people get vaccinated, it actually halts transmission. You're not going to get COVID if you have these vaccinations. We have all the vaccines we need. We just need our people to take it. A, for their own protection, for the protection of their family, but also to break the chain of transmission. You want to be a dead end to the virus. So when the virus gets to you, you stop it. You don't allow it to use you as the stepping stone to the next person.
Now we know that the vaccines work well enough that the virus stops with every vaccinated person. A vaccinated person gets exposed to the virus does not infect them, the virus cannot then use that person to go anywhere else. It cannot use a vaccinated person as a host to go get more people. That means the vaccines will get us to the end of this.
I want to point out that.
All right. So that's kind of depressing, I think. I want to leave with a positive note. The Journal of American Medical Association research said meta-analysis shows that individuals with higher level of optimism experienced a 35% lower risk of cardiovascular events compared to those with lower optimism, as well as a lower mortality rate. It went to the 2006 study. It says down here, it says optimists tend to live longer. So I want you to leave on the average from 11 to 50% longer than pessimists. So I want you to leave with the idea that your body is amazing. Your body is incredible. And because your body's incredible, you need to know that the things that you do for your body are important. That every day you exercise, every day that you do inward downward pressure on your flat bones, you're going to increase new blood production. The red blood cells are going to come, the new adult stem cells are going to come, the white blood cells are going to come and increase your immune system. If you just did 21 minutes daily of activity or 150 minutes weekly, you would reduce your all-cause mortality by 50%, according to the research that I showed in my first talk that I gave. You need to move the muscle groups behind the legs and the groin and the abdomen to milk those lymphatics and drain that poison out of your body. You have to reduce your free radical oxidative stress. Remember, free radicals are made from doing things like high aerobic activity, cycling, weightlifting. They all produce heavy amounts of free radicals in your body. If you're going to do those things, make sure you increase your superoxide dismutase, or the SOD. Of course, if you did Tai Chi and Qigong, you'd automatically be increasing only SOD and not having any free radical oxidative stress created in your body. But if you are doing those other exercises, make sure you eat proper food groups to be able to kind of find a way to clean your body out. Remember to laugh, sing, or to gargle to stimulate that vagus nerve. Use affirming words to activate your positive psychoneuroimmunology. Tell yourself you're loved and lovable. Tell yourself you're awesome. Give yourself hugs and let yourself know that you're worth it because you are loved and lovable. And I want to leave you with this idea that, your immune system's amazing. And we went through a terrible scare the last 3 years. And that scare had us do things that I think are just, as we look back at it, insane. Why would anybody take something that was 99% ineffective in stopping transmission that came with side effects for 1 in every 800 people? Like, why would anyone want to do that? Well, because we weren't told the truth. We weren't told the information. So I want to make sure you know the information. I want you to check it out yourself, know that it's accurate. You can find this information on my website at richardleirer.org. I will keep this PowerPoint presentation up. You can click on the sources and find them anytime you like. So thanks for listening. Thanks for watching. Thanks, folks and Toss, for coming in and being part of this new information about your immune system and your lymphatics. Thanks.